This guy was always a friend and a colleague and a mentor to a young faculty member who came in 1980. And Dwayne, who's sitting by me, equally so. When Guy left, Dwayne and I kind of managed this Kiracofe lecture series. And I really think it was kind of the brainchild of Dr. Davis when it comes right down where credit is due. And we kind of managed it. And it's been a thrill for us because we've had some really wonderful scientists come here over this last, gosh, it's going on 25 years or something, isn't it? So I chose to speak about something that I hope will be relevant to all of you. Rather than talk about something that I know a lot about, reproduction, I'm going to talk about something I know a little bit about. But I have read some literature on this and I found it quite interesting. For those of you that are students here, I hope this is informative to you to learn what the process of peer review is and what we do as scientists in trying to publish our research. And so I'm going to work here, Carol. I'll just give you a quick outline of what I'm going to do. It's kind of a collective group of things I'm going to talk about. Yeah, there it is. So I want to first give you a little bit of background and also my biases. We all have biases, and I think unless we recognize that, we're not being really honest with ourselves. So Carol mentioned I had 197 peer-reviewed journal articles during my 47 years as a faculty member and a graduate student in 6 different journals. The 3 years I spent as a section editor for that section of dairy science, now that is 3 different sections in dairy science. And so that was a mishmash of all kinds of papers that came in. Not a lot of them were reproduction and physiology. So it was really difficult as a section editor because there's so many things I didn't know a lot about as an editor. I handled 185 manuscripts in that 3-year period. I peer reviewed 40 years. And so my experiences and potential biases are evident except when I put citations on the slides. Okay, so I just want to warn you of that. So my aim is to help assist novice and even seasoned authors to publish their research and to help you understand the process a little bit. For students, this is going to be very new for you. For faculty, new faculty members, it's probably new for you because you learned this on the job. Nobody ever teaches you this in graduate school. I want to talk a little bit about research and the scientific method, qualitative and quantitative reviews and what those are, a little bit about peer review, the role of peer reviewers, what I think their responsibilities are, the role of an editor, an editor of a journal, and then a few things on writing tips for authors at the very end. So that's where we're going to go. I want to start with this dogma. Dogma is something that we believe is true. It's an established opinion, belief, or principle. It's usually based on some recognized authoritative group of thinking, but it's never been proved. It's not fact. Sometimes the dogmas that we believe in are so difficult to prove scientifically that we haven't done the right experiments. But I want to just give you an example. And for graduate students and younger faculty, I think there's a lot of dogmas we can challenge that would lead to good experimentation. So let me just give you an example of a dogma. Before 1950, medical students were taught that if you touch the heart, it would stop beating. Think about that for a minute. There weren't any open heart surgeries before about 1950. So what happened in research, they found if they added sodium chloride to the coronary arteries and changed the sodium potassium ratio in the blood, the heart would instantly stop. That means that they were able to do surgery after Russell Nelson and a couple of other pioneer researchers at the University of Minnesota developed the heart-lung machine so they could maintain cardiac patients on life support while they worked on the heart that was not beating. And then they found, obviously through research, that when the heart was exposed back to a normal sodium potassium ratio, that the heart would resume normal beating. And Russell Nelson was asked one time, from a cardiologist. He says, what if it doesn't work? He says, it always works. It is a scientific principle. It always works. And that's what led to open heart surgeries. The first were done at the University of Minnesota in South Africa. So that's a dogma that was later shown to be untrue by experimentation. So when we talk about reviews of the literature, there's qualitative reviews. That's what most of us are familiar with, where an expert is asked to make a scientific review of a certain topic. Okay, so that's the benefit, is they usually call on people that are really good, done a lot of work in an area, to author a review article. And they're helpful. Those qualitative reviews are helpful to other people to read them. They kind of give you the 10,000-foot level view of a specific topic, which is really helpful in helping you keep up with topics that may not be exactly in your interest area. And I have found, in my opinion, notice there's no, this is my opinion, best reviews provide what is known, but also what is not known or well documented. If a reviewer does that, he or she really contributes to the scientific knowledge, because it helps those of us that are not as don't understand those principles, or not aware of those principles, to know what really we don't know. So for a young researcher or graduate student, that would give them some ideas on, hey, this is what something that could be researched. Now, there's some cautions, I think, that we need to think about when we talk about qualitative reviews. Some of them contain biased views of the literature by the authors. We all have our biases. I've admitted mine. I've shared mine. Okay. And sometimes not all the available literature is reviewed. It's difficult to do that in fairness to reviewers, but it often highlights published literature of the author and their former students, I found. And I've especially noticed that in the last decade or so, going to professional meetings, when people are asked to do a review paper in a national meeting, they tend to ignore everybody's data but their own. And that's just not right. That's not fair to people upon whose shoulders we stand, that did a lot of really good research with not many techniques, not many dollars. You can go back 50 and 60 years ago. Now, there are quantitative reviews. These are generally called meta-analyses, because what one does is they combine and analyze data from multiple like studies, multiple like design studies, and they tend to remove some but not all of the author bias. So that's a good part. They give increased statistical power. So you have by bringing lots of data together, it gives you ability to give more robust conclusions, because you have a bigger data set. You're able to detect smaller differences, and they can contribute to the scientific literature. And they're comparatively low cost. I mean, you don't have any animals involved. It's basically your time researching the literature and perhaps contacting authors and getting some of their data, the raw data in some cases. So they're really add to the literature. And I've noticed in the last decade, at least in my discipline, we're seeing more meta-analyses being done. The cons or disadvantages, they do require statistical procedures that most of us are not very familiar with. In fact, some of our statisticians are not. I did a meta-analysis comparing the fertility of semen that was packaged in 1/4 cc versus 1/2 cc straws. And I had a hard time finding somebody in our statistics department that knew how to do a meta-analysis. But finally, I found somebody and we were able to do it. And so the other problem is a lot of us aren't familiar with those types of techniques. It's something that we haven't been taught. At least I wasn't taught. Maybe you graduate students now are being taught about meta-analyses and learn a little. No, I'm seeing some head shaking. So the validity of these meta-analyses really depend on the methodological quality of the data that of the studies that are being included, the eligibility of including certain studies or not including some studies, and of course there's the reporting biases that come with each of those studies. But these type of analyses, I think, have contributed a lot to what we understand. I've seen some really good ones done in my area in the last decade. So a little bit about the scientific method for a minute. You know, I looked up the definition of search and research just for fun. Search, the definition says you try to find something by looking or seeking carefully and thoroughly. I'm not very good at that. Ask my wife. Where's this? It's right there. And I went in there and it is there, but I didn't see it. Okay. I guess I don't search carefully. But research is interesting. It says it's a systematic investigation and study of materials and sources in order to establish facts and reach new conclusions. So research is better than search because you systematically go after solving a problem. So when we talk about the scientific method, which I would encourage my colleagues here and some of you students, if you haven't learned much about the scientific method, it's very valuable to do that. And perhaps we should spend more time teaching our students what that is. But the first thing it starts with is understanding what is in the literature. What are the facts? What are the theories? What are maybe some of the dogmas? What evidences do we have in a certain specific area? And then from that understanding, we can develop testable hypotheses that can be tested in an experiment. So we then would formulate objectives to describe what the experimental goals are of the study, create an experiment that allows us to test well-designed treatments with the appropriate controls. I could give you an example of something that Dr. Kiracofe dealt with a long time ago when he was doing his mastectomized cow research, because it was all based on denervated cows where their mammary glands were denervated, and they did an incomplete 2x2 factorial, and they left off one experiment. They didn't have calves nursing denervated cows. And if they had done that experiment like Dr. Kiracofe did with mastectomized cows, they would find out that those cows have long anestrus. But they didn't have that treatment in there. So people didn't believe the data until a guy came along and he had a hard time getting that research published because people wouldn't believe the data. And that's because the previous experiment ignored one important treatment. Scientific method involves making appropriate statistical comparisons, and those should be a priority. We should know what we want to test ahead of time to make sure that we are designing the treatments correctly, and then we need to properly interpret the findings, and that's not always easy. Some people overinterpret their data, and that's something we have to be aware of. You see in the literature that sometimes, and then we formulate the conclusions based on results, and then it starts a cycle. So I always just tell my students, a researcher's like a detective. You get some clues, you design an experiment, you figure something out, and now you got 6 new questions you could ask. And good experimentation leads one experiment to another. Okay, a little bit about experimental design. The implications of one experiment. Results of one study conducted at one location should be cautiously applied to the entire population. So this is what we learn in statistics. We got a population of experimental units. We're going to sample the population, hopefully by getting a random sample. And then we're going to generate some data, some means. And we're hoping that the average there is going to match the average of the entire population. If we do the experiment right, get the right number of animals, we have they're randomly assigned and so forth, then we can make inferences from that sample to the population. But here is the problem that I've seen among some of my colleagues in my discipline. The caution is one study can prove a physiological principle to be true, but that should be further tested at multiple locations. Because it may be that that given site is unique and those experimental treatments may not be replicated and repeatable in other sites. And so I think we need to be cautious and careful about that. So the experimental unit should be representative of the entire population. So in my area, dealing with reproduction, there's a difference in the fertility of primiparous versus multiparous cows. On probably 99% of the dairies, fertility is always going to be higher in a first lactation cow than an older cow. Okay, I'm going to pick on my dairy nutrition colleagues. This has been violated in numerous dairy nutrition studies when they do feed intake studies where they're getting individual feed intake, because they always use, almost always, only multiparse cows. Now, their rationale is that they want to assign the experimental treatments to the treatments, the experimental units to the treatments, and have as much information as they can, and they have a first lactation information. That's important because they're going to measure milk production. So they use multiparous cows because they already have a lactation information. But here's the problem. The proportion of primiparous cows in commercial herds today in the dairy, about 25 to 40%. Some of the large dairies, it approaches 45, sometimes 50%. So they're taking information from a population of multiparous cows and applying it across to dairies that have lots of primiparous cows. That doesn't work. I tell them this, but they don't listen to me. That's another thing. All right, so let me give you an example of what Food and Drug Administration requires. We go back and talk about bovine somatotropin. The foundation studies that were done at Monsanto in over near St. Louis, they started out with 4 different doses, intramuscular doses. They were tested in 28 primiparous cows and 57 multiparous cows. Why that? Why with the number of cows? Because that was 30% of the cows were primiparous. That matched the industry, okay? That's an important point. So they took that information and they did a proof of concept study where they compared the control to what they found to be the minimal effective dose. They did that in 4 locations. Notice where these are: New York, Utah, Florida, Arizona. 4 different regions in the country. And they did that in 92 primiparous cows and 272 multiparous cows. Then they went back and said, oh, maybe we don't want to give intermuscular injections because we'll have carcass blemishes injecting in the muscle. So let's see if we can do it subcutaneous and use the ischial tail head area where there's not a lot of carcass meat used from, okay, and see if that works. So they went back to the beginning, did a foundational study, 21, a third of the cows were primiparous, compared a 500 milligram dose. And they had both intramuscular and subcutaneous. So then they took that to the field, went back to those same herds, 4 different locations, and they did a dose titration study again. Then once they found that 500 milligrams worked, they went to what's required by Food and Drug Administration, multiple locations, 28 herds, 1,213 cows, the 2 doses in 7 herds in each of the regions of the United States. That's what's usually required for an FDA study. So that is an example of really thoroughly following a pattern of experimentation that one should do. And that's not always something we can do at a university unless we team up with colleagues at other universities and use similar treatments in different locations, which I did in my career, both in dairy and beef. So a little bit about peer review. I was surprised when I read this paper that peer review was accepted by most biomedical journals by the late 20th century. That was during my career. I mean, some of these biomedical journals were not using peer review before 2000. I was blown away by that. So what's the purpose of peer review? Is to help the editor. Okay, the editor has been selected. The editor needs help in determining suitability of a manuscript, whether it's worthy of publication. Does it offer new knowledge and so forth? We'll get into that in just a minute. So the editor assigns usually 2 or more experts, okay, in quotes, to review and provide positive feedback on scientific merit. So after they review the paper, they suggest to the editor whether this paper should be accepted without revision, accepted with revision, so it'll go back to the authors. Resubmission is an option where they say, well, it's a good paper, but it really needs a bunch of work done on it. So they send it back for a really complete revision, and then we'll re-review it or just reject it. Okay, that's what reviewers do. They provide recommendations to the editor. Some reviewers think they are the editors, by the way, responsible. That's not the case. Peer review remains the accepted quality control process involving blinded or unblinded scrutiny of manuscript merit. That was the author's solutions. So let's talk for a minute about blinded and unblinded. Blinding is a form used for closed peer review. Now, most of us are familiar with that and published. Single blind means that the reviewer, the editor doesn't know who, I'm sorry, the authors don't know who reviewed the paper. That's single blind. Or it could be that it could be also that the identity of the authors is not known by the reviewers. Now, when I started here in 1980, we had 5 poultry scientists, and the journal Poultry Science was double blinded. So they would tear the cover sheet off the manuscript, so the reviewer didn't know where it was coming from. That's actually single blinded. The reviewers didn't know the authors. It was double blinded because the authors didn't know the reviewers too. Okay, so double blind means that the reviewers and authors are not aware of each other's identity. And so that's what Poultry Science did. I don't think they still do today. I don't see Scott. I was going to ask him that, but I don't think they do that anymore in Poultry Science. Triple blind would include the editor not knowing. So the editors, the reviewers, and the authors don't know who each other are. And of course, that's claimed to be best to prevent bias, but I don't know if anybody does that. It gets really difficult when you do a triple blind type of thing. So let's look at the literature here. A meta-analysis was done in 2022 where they concluded that double-blinded peer reviews were associated with 18% fewer manuscripts being accepted. So the authors and reviewers didn't know each other. So the acceptance rate was down in this meta-analysis if it was a double-blinded reviewed paper. And the second paper, they said some evidence that double-blinded reviews can mitigate reviewers' bias towards the authors. And that's an issue. You know, sometimes reviewers, they get a paper to review, they may not like the person, or they're a competitor, or, you know, there's lots of things. So a job for an editor to choose the proper reviewers is really critical to make the process fair to everybody. Now, what are some of the biases that have been identified in this paper here of peer review and reviewers? Narrow-mindedness of reviewers. I learned a long time ago that if I didn't like what the reviewer said, it's my fault that I didn't do a very good job of explaining. Then I would get mad, Mike. Because if they didn't understand something, I didn't communicate very well, and you should accept that as an author, okay? But there are some people that are narrow-minded. They probably shouldn't be reviewers. There's a reluctance of reviewer to accept groundbreaking and innovative research. That's what these authors concluded. Some people just, you know, if it challenges the dogma or the scientific evidence, they're reluctant to accept it because it doesn't, you know, it doesn't fit the model. Then there's the Matthew effect, what they call the Matthew effect. That's where a reviewer submits a manuscript, supports a manuscript that's based on the author's reputation rather than the quality of the manuscript. And I will tell you, as an editor, sometimes some of the most well-considered colleagues that were publishing had some of the worst manuscripts. So that's the Matthew effect. We just don't, it doesn't matter if it comes from so-and-so who is really considered to be a leader in the field. If it's a poor quality manuscript, treat it as such. Be fair with them, but treat it as such. So having reviewers increases turnaround time. Now everything's digital. Back in the early days of my career, everything went through the mail. And I remember an editor showing all the places manuscripts went back and forth between editors, reviewers, authors, and finally gets to the publication stage. You know, it took a lot of stamps. Some reviewers are very non-constructive and over-positive, provide over-positive, non-critical reviews. They like the person, and so they're just throwing flowers at them where they really probably shouldn't. A lack of adequate or willing reviewers. Now, I know someone in my field who will not review papers. He's a leader in my field, and he refuses to be a reviewer. To me, that just is not right. We have the obligation to help each other. So and it's hard to find the right reviewers, the right experts. Inability of reviewers to detect shortcomings in methodology, statistics, and interpretation. I've seen that more and more. More and more papers I'm still reviewing papers. I get one or two every month. A lot of authors do not understand statistics, and oh boy, it's a mess. Some of the things that we see. Reviewers' role, these are my opinions. Does the manuscript contribute new knowledge? Does it report and contribute essential confirmational data? Okay, here's getting back to what I said earlier. You could have two papers, and this has happened to me a number of times. I was doing research similar to what somebody else was doing. They published it 6 months before I got mine published, and the reviewer says, "Oh, that's already been done." So in my response to the editor, I says, these experiments were going on simultaneously. I didn't know this information when I designed this experiment. So you have to think about that a little bit and remember that there is need for confirmational reports. They may not be similarly designed, but that goes back to the important point of inferences coming from more than just one population or one sample of the population. We need to make helpful, non-condescending suggestions. I have received reviews, like many of you may have received, where the reviewer just talks down to you like you're a dummy. You know, that's just not right. That's not fair. They shouldn't do that. If they don't want to be helpful, they shouldn't be reviewers. Then we should have positive suggestions. And I always try to do that, whether it's grammar or anything else, because I want to help that person. And that was always my opinion as a reviewer. This was Bob, oh geez, I just lost his name. He was Ernie Minton's mentor at Wedeman, thank you. Bob Wedeman, who was a university distinguished professor at Oklahoma State. We had this discussion when I was a young faculty member. We had Michigan State in our background, and he says, my role is to help people get their data published, unless there's something wrong with design, you know, obvious things like that. But I should be able to help them because I want them to help me. And I think that's the right attitude to have. So what about editor versus author nominated reviewers? Most journals now, they ask you not to provide reviewers that you would like to review the paper, and those you don't want to review the paper. Okay, that may be important too, because you know somebody doesn't like you out there. And so what happens is there a favorability for success? Well, although review quality scores didn't differ in this study, they found that author nominated reviewers used by the editor actually on first review accepted with revision more papers, or recommended exception or revision of more papers than if the editor nominated the reviewer. Okay, so the idea is you're nominating somebody that you think will be fair to you. It's probably somebody you know well, and it looks like there's some bias when the editor uses an author nominated reviewer rather than the editor choosing somebody off the editorial board who may or may not know you or whatever. But when it came to the final review, there was no difference. So after revisions and so forth, there was no bias at the final review. Now, when it comes to rejection, the author nominated reviewers rejected fewer manuscripts. That was their recommendation, but upon final review, there was no difference. So there seems to be a little bit of a bias there when the editor uses an author-nominated reviewer. Here's another study showing quality scores were not different between author-nominated reviewers and editor-nominated reviewers. Days to review completion, in other words, they took about 3.5 weeks before the review came back. But the author-nominated reviewers recommended acceptance more often than revision or rejection, and that's the relative risk. So 50% of the time, the author-nominated reviewers would recommend acceptance or acceptance with revision. So that's something probably an editor needs to keep in mind. relative to who he chooses or who she chooses as reviewers. Here's another data set. This is from Journal of American Medical Association. They did 780 reviews of 329 manuscripts in 10 different biomedical journals. And the reviews were equal quality between reviewer types. So didn't matter if the editor chose the author nominated reviewer or the editor chose the reviewer. The quality of the reviews were the same, but the author nominated reviewers were 1.64 times more likely to recommend acceptance. And they were also 2.66 times more likely to recommend revision. So I think you see what the literature's saying, that the author nominated reviewers are going to be a little bit more favorable to you. So I guess the word to the wise is that choose people carefully. I mean, if the editor chooses to use one of your suggestions, choose somebody that's going to be fair, somebody you think will be open-minded, and that may be helpful to you in getting your data published, as long as there's not any real deficiencies that would prevent it and call for rejection. So, editor's role. Editors must be editors. The last 15 years, I have seen this more and more, where editors keep sending back to the reviewer again and again the same paper. I see some of you shaking your heads. That just isn't right. Editors have got to make a decision. They can't let the reviewers make the decision. That's their role as an editor. They have to make that decision. And I know there's one of my colleagues who's not here right now. He refuses to look at a paper twice because they keep sending it back to him. And so I think that's, if you're going to be an editor, you need to be an editor. You got to make the tough decisions. And I had papers where I got a split decision between the two reviewers. In some cases, the editor felt he or she was capable of serving as a third reviewer and did so. And it helped. In that case, that paper got published. So I think that's important to keep in mind in terms of how we handle things as editors. We need to protect confidentiality of authors and reviews. If we don't do that, we're going to destroy the system. If we're talking behind our back, if you go up and say, I reviewed this paper for you, that's inappropriate. You shouldn't do that. It's just not, it violates the confidentiality of that process. Editors got to choose good reviewers, good, fair reviewers. That's really critical. They need to make decisions with a reasonable amount of speed, not sit on things for a long time, and make sure they communicate back to the authors exactly what the standards are of the journal and what really needs to be revised. Sometimes you get a paper back and you're really not sure what, you get the authors, the reviewer's suggestions, but the editor doesn't give you any real feeling for where you should go with it, because you could get two diametrically opposed reviews. You see lots of heads going up and down. And so what do you choose? If you choose to revise according to this reviewer and not this reviewer, you probably just got yourself in hot water, depending on what this editor does. And so editors have got to be very careful about how they do that and be fair with people. Author tips. Carol's got up here something left for you. I'm going to pick on my swine colleagues. Will work as food. Due to this grammar mistake, Wilbur found a position. It just wasn't the one he wanted. Okay? So we need to communicate what you really mean, and the consequences are something that you are not expecting. Okay? It's really important. This next statement from Lowell Brandler. Guy, remember him? He was the editor, the Kansas Agricultural Experiment Station editor when I came in 1980. I sent a manuscript over. In those days, you wrote a manuscript, you typed it, your secretary, your office professional, I'm old school, sorry. Your office professional typed it up. They sent it over to the editor to get a contribution number. And before that, somebody in the department had to review it. We had that policy. Then it went to the editor, and Lowell had a PhD in communications. he actually, I went back and looked at his history online, and he actually had PhD students while he was the editor. And he had read my paper and used a lot of red pencil. And he had typed up this little thing and put it up and taped it on the back side of one of my manuscript pages. And it said essentially, good writing occurs when it not only promotes understanding, but prevents misunderstanding. Boy, that really stuck with me. That really helped me think about my sentence structure and how I would state things. I remember in statistics, a small bird study or a study of small birds. It's two different things. So you've got to communicate correctly, right? Okay. Proofread your submission multiple times. I mean, how many times as a reviewer and editor, I get a paper, there's misspellings in it, all kinds of stuff. It looks like, and sometimes the abstract didn't agree with the data in the tables because I think as they were putting things together, they forgot to go back and change the abstract after they did the revisions. Be really careful. That's what spell check's for. It works except in my area because diary and dairy never gets right. Okay. Be meticulous in your description of design and methods. Because the purpose of a methods is for somebody else to actually repeat what you did. And if you don't provide all the methods necessary, they can't do that. Okay. Statistical procedures should include initial tests for homogeneity of non-discrete outcome variables. That's why we have transformations. Do we test for that? Do we test for, remember, analysis of variance is very robust, but it requires homogeneous variance and randomly assigned treatments and so forth. And you need to, especially in my area where we were looking at hormone profiles, very often we get heterogeneous variance. And so we have to test for that and use a transformation. Now I've noticed in the last few years, people are asking if you did an a priori sample size calculation. Now that's important to you because it allows you if you're not using a bimodal outcome variable, you know what the variance is. You can predict how many experimental units you need to assign, so you're not over assigning treatments or under assigning. Under assigning is just as bad as over assigning because then you end up with type 2 errors where it looks like it's different, but you don't have enough power to detect the difference. And so that it's really important in my area where you're using a bimodal variable, pregnant or not pregnant. If you want to detect a difference of 5 percentage points in the 45 or 50% range, you got to have about 800 counts per treatment. And so that's really important to do. Statistical descriptions should include model components. When I read the paper, I want to know what was in the model. I've read papers that don't give you that information, and they talk about differences in multiparous cows and primiparous cows. Was that in the model? I mean, they didn't even tell us that. Here's when it's violated the law. Treatment results are never similar. Statistical tests test for differences, not similarities. So two treatments do not differ, they're never similar. And I see that all the time. Remember what you learned in statistics. And this happens often. Never duplicate result data in the text that's already in a figure or a table. Don't unnecessarily duplicate data in your papers. That's important to do that. Well, I'm going to conclude there. This Happy AI Breeding is my sign off for my 298 artificial breeding monthly contributions I did in Hordes Dairyman between 1987 and 2022. I think I got that job because the editor was a K-State grad from Topeka. But I was only here 7 years when he asked me to do that. And that was one of the most enjoyable, difficult jobs I had because I had basically a monthly column every month for all those years. I had to come up with a new idea. But nothing, I'll tell you, nothing was more rewarding to me as a person when I'd go to a conference, particularly a dairy conference, where I'm talking to veterinarians and dairy producers, and they'd come up and tell me how much they love to read my columns. that really meant a lot to me. Because essentially my audience was digital, and where that magazine Horse Dairyman went, which at one time had 130,000 circulation throughout the world, is translated in Japanese and Spanish. That was just a thrill for me. You know, once in a while, a student would come up to you and tell you they enjoyed your class. That was always nice, but really, when I was trying to do research to help dairy producers, that was a thrill for me when I get a compliment there. So thank you for your attention.
Time for a few questions.
Mike?
Yeah, I have a question for you on, one area I guess you didn't talk about was the proliferation of journals today, how many more we have today than when you started your career.
Yeah.
And how that's influenced publication.
Yeah, for those of you who didn't hear his question, he's talking about the proliferation of journals. And I might add to that proliferation of authors on every journal article, which has caused journals for you to indicate what each author did in that paper. I think that's a problem when there's more and more journals. I don't know why we need more journals. It makes it more difficult for us to keep up with the literature. You know, dairy science has done it. They got JDS Communications. Animal science has done it. I'm sure other sciences have proliferated that, and it makes it more difficult. That is a challenge. It's really, really hard as a faculty member to keep up with research when you have extension and teaching responsibilities and everything else. It's a challenge, and that's a good point you're making. Yes.
There are some like reviewers who want their view to be written again and again by other authors so that they validate their theory. And that's why I think sometimes they push back when someone challenges his theories, so that his theory, the legacy of his theory remains for quite a long time. So how do you think the authors should respond to those concerns?
Well, the question is responding to reviewers' criticisms for various reasons by the authors. I didn't explain this for the graduate students, but when a reviewer reviews a paper, they give you a list of suggestions. And you have to, for the editor, respond to every suggestion when you send back your revised manuscript. And you just have to be honest and tell them why you did what you did and the reasons you did what you did. And I think if an editor is reasonable, he or she will accept the honest responses and they will be fair to you. You can't hide things from people. If you try to hide something, good reviewers will pick things up. I'll tell you, since I've been retired for ample time, but when I do a review, it takes me a better part of a morning or afternoon. And it's usually about 3 pages long. I do a thorough review because I want to help the person, and a lot of the reviews I get, people are writing in a second language. It's not English, is not their native tongue, and so you can't hide things if somebody reviews it carefully. So you just have to honestly respond to them and do the best you can so the editor knows where you're coming from, and then hopefully he or she will be fair with you.
I have a
Yes.
question.
Go ahead.
Besides getting mad, what is your advice for the first thing to do when you get rejected on your paper? Person.
Let it set a while. And come back to it. That's a good reason when you write a manuscript, let it set a couple days and then come back to it. That's a real good writing tip. I got really mad one time. This was way into my career.
Only one?
You're keeping me honest. I said really bad. It was a paper where we had looked at LH surges, LH release in cows that had synchronized. We had 170 cows in that study. And it was reviewed, I think, by somebody from Australia or New Zealand, where timed AI was not very acceptable at the time, and they saw no value in the paper whatsoever. So even before I answered the reviewers, so one reviewer was just terrible. And one reviewer was a fair review. I called the editor and I said, look, I'm gonna withdraw this paper. I'm gonna send this to another journal. This one reviewer is just totally off base. And the editor says, you're right. Just ignore that review. And let me go forward. Now, that was the editor's decision, so I had his review and the other reviewer had 2 reviews. But he let me go forward, but it really made me mad. This person just didn't understand what we were doing and was just overly critical. And I'll tell you, in the 25, 30 years ago, timed AI was not well received outside the United States. It really wasn't. I had a colleague that was very well respected in Ireland that just thought what we were doing was crazy. But we wanted him over finally, and he saw what we were doing. I showed data this morning from 20 to 24, 2020 to 2024, 6 months after Victor came. And our first service fertility and time insemination in dairy cows is averaging 50% in our dairy. And we're using the right tools. We got good people doing the work. So it's changed the industry. So it's important. So, I don't know if I answered the question, but that's what you have to do. You just kind of have to let it go sometimes. And take responsibility if you didn't communicate correctly. That's what you're gonna have to fall back on. Sometimes we fail to communicate exactly what we want to say.
I have one here.
So Jeff, I see AI up there. I was in a presentation last week. It was all about ChatGPT, artificial intelligence, and how that is being used these days. And I'll get my numbers wrong, but you can scan in 5,000 pages of PDF and lay out what you want it to address, and it will spit a paper back to you. And I'm thinking about discussions and introductions and those kind of things. How do you think that'll affect this process?
Yeah, so Dr. Day's question was about, AI, artificial intelligence, and writing papers. I did read one paper when I was doing this at PubMed that said that reviewed all that area and said in the balance in the end, we're still gonna have to have people read the paper, make decisions, and determine if it's real or not. So it's gonna come back to competent people, even though that's the. So there's a lot of information in PubMed. I just shared a little bit with you. There's quite a bit of information on some of these things that I talked about today. If you have any interest in them. Yes, Bob.
Jeff, 2 comments, or 1 question and 1 comment. What's the fate of peer review as animal science disciplines shrink? For instance, I was always told in the poultry industry, there are 2 universities, and you get one review and they get your paper, and how is that gonna affect peer review?
Yeah, so Dr. Goodman's asking about peer review as faculties are shrinking. We have fewer people that are working in particularly poultry areas, some areas, maybe horse areas as well. That's going to be difficult. I don't know, somebody said one time, peer review is the worst thing that we have, but it's the best thing we have. And I don't know how we're going to get around it. How else, unless you want to unilaterally let editors make those decisions without any input from the outside. I hope we don't come to that, because you're gonna have a hard time finding editors that are well versed enough in many parts of even one discipline to be fair and accepting, and nobody's gonna want to accept that responsibility, because that means you're gonna have to read meticulously every paper. And I'll tell you, as an editor, when I got the reviews back from the reviewers, then I read the paper. Okay, then I sent to the author my suggestions on the PDF and said, this paper has been recommended for acceptance. If you consider these following suggestions. And so they've already answered the reviewers. I'm asking them to look at my suggestions. And I remember one of my colleagues, he's a friend of mine, very, very proficient. And I won't say his name because a lot of you know who he was. Had one of his papers, and their abbreviations were not mnemonic. Mnemonic means that the abbreviation says what the treatment is. In fact, the treatment designations were confusing. They overlapped with one another, and I suggested that they change. Oh boy, I get blasted on that thing. Not only him, but his colleague took me to task later on in the meeting. And it didn't, it wasn't right. So I think more eyes that see it, the better. And that's gonna be a problem in the future. I just hope we don't allow just editors to make those decisions. You had a comment.
My second comment, I calculated it. I was certainly not as long as Dwayne, but I was across the hall from you for 5% of your career. And I enjoyed it very much.
No, that's nice. I appreciate that. Maybe if it was more than 5%, you would have.
You brought up the name Lowell Grinder, who was the editor of the Ag Experiment Station. Brought back some memories to me. Early in my career, I had him review an article that I wrote, and I had about a 3-sentence paragraph. He marked the whole thing out and said, none exhibited asterisk. That made me conscious of being worded.
I was a graduate student at Michigan State University. Ron McGillier, who was an apple breeder, there was a bunch of offices of graduate students together, and he had a little corner office with an architectural table, the ones that would go like this. That's where he did all his editing. He was editor-in-chief in Journal Dairy Science for years. And he could take what you're saying here, he could take a 15-word sentence and say it better in 9 words. That's how good an editor he was. So when you get to a point where you can really write well and succinctly, I can only say the word, you've mastered the language. And so I think that's a talent that people acquire. And you can become better writers as you do more of it. I really think you can.
I have a question here, Dr. Stevenson.
That's Musa.
Is that you, Musa?
Yeah, that's me. So first off, I just want to extend my gratitude to Dr. Stevenson for all he's done for us as graduate students. Some of us are actually in our PhDs, and we are actually learning from some of the skills that he's taught us during our master's program back at K-State. Just a quick, I just want to get your thoughts or your suggestions on, so as part of my PhD program, one of the trainings that we actually get to, do here is that, our advisors share, review papers. So they are actually, reviewers for, a lot of journals. So a lot of times they share those papers with us as graduate students to, give our insights, and then we meet and then discuss. Sometimes it can be very intimidating when you see authors, for instance, of your caliber. You as a graduate student, you are, kind of reviewing publications of, of high status authors like you. What suggestion do you have to share with us? To be able to kind of like mitigate or control that intimidation. I understand.
What?
How to not be intimidated?
Oh, how to be intimidated? I don't know. I always thought I was a nice person. My wife says I intimidate people.
I think he's asking about how not to be intimidated when you're reviewing a paper from.
Yeah. Now, it's a really good question. I think it's important to take and value what's in the paper and disregard what I talked about was the Matthew effect. Don't think about who wrote it, just read it for what it is. Because the Matthew effect is unfair. If you just think that so-and-so writes, always does good things, but the paper's not very good, you have to treat it as such. So just treat everybody the same. That's the golden rule, isn't it? Treat everybody the same regardless of reputation. I think you won't be intimidated.
All right, sounds good. Thank you.
With that, let's thank Dr. Stevenson. Sure appreciate it. Sure appreciate you, all you guys attending, and thank you very much.
Thanks for coming.
You can read the paper without reading the authors.
Yeah. Well, that's where the blinding comes in.